For decades, we've basically been sold this idea of the medical silver bullet. I mean, you know the story. You find the single rogue protein that's causing a disease, you design one perfect chemical to block it, and boom, the patient is cured.
Right, it's a very comforting narrative.
Yeah, it's clean, it's linear, kind of like a sniper taking out a target. But that linear approach is almost entirely a myth. In fact, that single target thinking is exactly why something like 90% of clinical drug trials ultimately fail.
It's staggering. And that failure rate is really a stark indicator that we've been treating human biology like a simple machine with isolated parts, when the reality is that we are looking at a highly entangled, totally dynamic system. When you try to hit one target, the system doesn't just stop. It adapts.
Which is why today we are taking a deep dive into a massive paradigm shift in modern medicine. We're pulling from a really comprehensive review published in partnership with the Hormel Institute at the University of Minnesota.
Yes, the paper is titled AI and Network Biology for Rational Polypharmacology in Signaling Drug Design.
Right. And our mission today is to unpack how scientists are finally abandoning that doomed silver bullet approach. Instead, they are using artificial intelligence to intentionally modulate entire biological networks. It is a shift that's, it's turning incurable diseases from biological mysteries into solvable math problems.
Okay, let's unpack this. Why exactly does hitting one target fail so often?
It really is a fundamental rewriting of how we discover drugs. So traditionally, pharmaceutical companies rely on what we call target-based screening. They simplify complex biology down to a single assay test in a lab.
So just isolating one protein in a dish.
Exactly. They find a chemical that effectively blocks an enzyme in a petri dish, and they think, great, we have a drug. But when that chemical enters a living organism, the surrounding biological network compensates for the blockade.
Because biological processes aren't linear pathways, right? They are these massive interactome graphs.
Right, an interactome is a web of genes, proteins, and metabolites that are constantly communicating. So if you blindly inhibit a hub protein, and a hub is one of those critical nodes holding the whole network together, the system doesn't just sit there and accept it.
No. Not at all. It reroutes around the blockade, or worse, the whole system crashes. And that is exactly where those massive off-target toxicities and drug resistances come from.
In network biology, they don't even see diseases as a broken part, do they?
No, they are visualized as a network failure. Specific clusters of disease genes, which we call modules, they get partitioned together and become dysregulated.
Okay, I like to use a visual analogy here. So if a disease is a massive traffic jam, a traditional single-target drug is like putting up one giant roadblock on Main Street. You block the road hoping to stop the jam, but the traffic just reroutes.
It spills over.
Right. It spills over, clogging up all the smaller residential streets. So the solution proposed in this paper is rational polypharmacology. Instead of putting up a roadblock, you are timing all the traffic lights across the entire city simultaneously to get the traffic flowing naturally again.
That's the exact mechanism at play. You are intentionally adjusting multiple targets at once. Historically, hitting multiple targets was considered a bad thing.
Oh, really? Why?
Well, pharma used to call it promiscuity in a drug, because it usually meant highly unpredictable side effects. But network biology turns that bug into a feature. We can now design a single drug, or a specific combination of drugs, whose unique topological fingerprint in the network shifts the entire disease module safely back to a healthy state.
Okay, before we move on, let's just do a quick 5-minute recap for you listening. So traditional single-target drugs fail because biology reroutes around them. Network biology views diseases as complex webs, and this new approach, rational polypharmacology, hits multiple targets to safely reset the network.
Precisely.
So a question for you listening to mull over. Think about a time you tried to fix one small habit and realized it affected your whole routine. What is a hub in your own daily network? Okay, but wait. Timing one traffic light is easy. Timing 1 million of them perfectly so you don't cause a 10-car pileup seems, impossible.
For a human, yes.
Right. The human brain cannot possibly process the millions of protein interactions required to know which multiple targets to hit simultaneously without causing severe toxicity.
You're absolutely right, which is why this field was stalled out for so long. To do rational polypharmacology, you need biomedical knowledge graphs. These are massive databases, things like HISHINET, String, Reactome, and KEGG.
And what exactly are those mapping?
They map multi-omics data. They link genes to proteins, proteins to metabolites, drugs to diseases, and even drugs to specific side effects. But to actually read that map and calculate the timing of those traffic lights, we have to rely on artificial intelligence.
Let's dig into the how there, because AI is such a buzzword right now. How does an algorithm actually ingest a biological network and spit out a multi-target drug? The source mentions a process called a teleport-guided random walk. What is that actually doing?
So imagine a digital scout exploring this vast biological map. The algorithm drops this scout onto a specific node, say a disease gene. The scout starts walking along the edges of the graph.
The edges being the biochemical relationships between things.
Exactly. Moving from a gene to a protein, then to a drug that interacts with that protein. Most of the time, it follows direct biological links.
Oh.
But occasionally, to prevent getting stuck in a local neighborhood, it teleports.
Teleports, like it just randomly jumps.
It jumps to another node based on semantic similarity, rather than a direct physical link.
Oh, okay. So it's gathering thousands of these random long-distance pathways to map out how different biological neighborhoods relate to each other.
Yes, it generates massive sequences of nodes. But sequences are just lists. The AI needs to do actual math, so it uses a mechanism called a heterogeneous skipgram model.
Heterogeneous skipgram, that sounds intense.
It's the critical translation step. It takes those exploratory sequences and translates them into what we call an embedding space.
An embedding space. So it's taking a complex, like 50,000 dimensional biological reality and compressing it into pure geometry.
That is the perfect way to visualize it. It compresses the biological entities into low-dimensional mathematical vectors. Once a drug and a disease are both represented as vectors in this geometric space, the AI can measure the physical distance and the angle between them.
You know, this sounds exactly like how a social media algorithm works.
How so?
Think about how a social network knows two people should be friends. Even if they've never met, the algorithm sees they share mutual friends, they check in at the same coffee shops, they like the same posts. The AI converts all those behaviors into math, realizes their vectors overlap in geometric space, and flags them as a match.
That's a great analogy.
So this AI is doing the exact same thing, but playing matchmaker between a drug's multi-target profile and a complex disease module.
And just like a social media algorithm gets better with more data, these drug discovery models use advanced algorithms like XGBoost classifiers to analyze those overlapping vectors and spit out a precise probability score.
Like it might say there's a 93% chance this works.
Exactly. There is a 93% probability that this combination of existing drugs will stabilize this specific cancer network.
But the AI isn't just matching existing drugs, right? The paper goes much deeper into deep learning frameworks. It mentions graph convolutional networks and generative models like diffusion networks. Those aren't just looking at old drugs.
No, they're creating entirely new ones. A graph convolutional network, or GCN, doesn't just look at the list of connections. It analyzes the actual topology, the shape and physical structure of the biological pathways.
Wow. And the generative models?
Generative AI models, like the diffusion models people use for AI art, they start with chemical noise and iteratively refine it.
Yeah.
They literally hallucinate entirely new molecular structures from scratch.
Hallucinating new drugs, that's wild.
And they optimize them to bind to 3 or 4 specific proteins simultaneously without touching the hubs that would cause toxicity.
Okay, let's do another quick recap of where we are. So AI uses knowledge graphs to link all this crazy biological data. Teleport-guided random walks and skipgram embeddings turn biological relationships into geometry, into math. And generative AI can actually build new multi-target compounds from scratch.
Very well summarized.
So for you listening, think about this. If an AI mapped your personal knowledge graph, what random nodes, say, your favorite food and your sleep schedule, might it connect? Okay, moving on. It's incredible that AI can map these connections in a vector space and hallucinate a custom molecule. But a mathematical hallucination isn't a biological reality.
Not at all.
A computer saying, I designed the perfect multi-target drug, doesn't mean it actually works. What happens when these perfect digital molecules make contact with a messy, living cell?
That brings us to the most crucial phase, the closed-loop workflow. You cannot do AI drug discovery without the wet lab.
The actual physical lab.
Right. The workflow starts with the AI model generating the hypothesis, but then it immediately moves to experimental validation, followed by pharmacokinetic modeling, and finally, clinical translation. It is a constant loop of physical feedback refining the digital algorithm.
Let's talk about that experimental validation, because proving a drug works on multiple targets simultaneously sounds incredibly difficult. The source mentions thermal shift assays, like CETSA and nanobret. How do those actually prove the AI's hallucination is real?
Think about thermodynamics. When a drug physically binds to a target protein, It stabilizes that protein's folded structure. CETSA stands for Cellular Thermal Shift Assay.
Okay, how does it work?
Scientists take living, breathing cells, introduce the AI-designed drug, and then literally heat the cells up.
Wait, they are trying to melt the proteins.
Exactly. Unbound proteins will denature or unfold at a certain temperature. But if the AI's drug successfully engaged its targets, those specific proteins will become more resistant to heat.
Oh, that's clever.
Yes. By measuring which proteins survive the heat, scientists can prove that the drug physically engaged with the exact multiple targets the AI predicted inside a live cell, rather than just in a computer simulation.
Okay, but just because a drug physically sticks to a protein in a cell doesn't mean it's actually curing the disease network. It could just be a useless, heat-resistant roadblock. How do they prove that hitting those specific targets actually shifts the disease module back to a healthy state?
That is where they have to prove network causality. To do that, researchers turn to CRISPR technologies, specifically techniques like perturb-seq or crop-seq.
CRISPR, right, gene editing.
Exactly. This is where biology meets big data. They use CRISPR to edit or mutate multiple genes simultaneously at the single-cell level. By intentionally knocking out the specific hubs the AI targeted, they can sequence the RNA of that cell and watch how the entire transcriptome reacts.
So they're essentially recreating the drug's intended effect genetically to see if the network responds the way the AI's math said it would.
Exactly. If the transcriptomic shift matches the AI's prediction, you have physical proof that the network map is causally correct.
That is brilliant. But again, a single cell in a petri dish is not a human being. I mean, the immune system, the liver, the blood-brain barrier, none of that exists in a single cell assay.
Which is why the loop must progress to translational models before it ever touches a human. This is where patient-derived organoids, or PDOs, come into play.
Patient-derived organoids?
Yes. Researchers take a biopsy of a patient's actual tissue and grow a miniature 3D version of their organ in the lab.
Okay, so PDOs are effectively biological crash test dummies built from the patient's own DNA.
That's a great way to put it.
We're crashing the AI's drug into a mini version of their tumor or their liver to see what breaks before we ever put it in their actual body.
That is highly accurate. You were looking for emergent behavior. How does the multi-target drug affect the 3D tissue architecture? Does it trigger an immune response? And even after the crash test dummy survives, there is one more step before clinical trials, MIDD.
Which stands for?
Model-informed drug development.
Right, because knowing the drug works doesn't tell you how much of it to give a patient.
Exactly. MIDD uses complex pharmacokinetics and pharmacodynamics mathematical models, PKPD. It calculates exactly how the drug will be absorbed, distributed, metabolized, and excreted by human body. It bridges the gap between the biological signal seen in the organoid and the precise safe dose required for a human clinical trial.
Are there any real-world examples in the paper where this closed-loop system has actually worked?
Several, actually. In oncology, deep learning was applied to the PI3K-AKT-MAPK network.
Sounds complicated.
It's a notorious, highly resistant cancer pathway. The AI found optimal dual inhibition pairs that shrunk tumors by hitting compensating nodes simultaneously.
Wow, avoiding the severe toxicity of traditional chemo.
Yes. And in neurodegeneration, machine learning analyzed transcriptomics to identify specific neuroinflammatory hubs like MAPK and MFB. This opened up new multi-target approaches for Alzheimer's and Parkinson's that were previously considered untreatable.
Let's do another quick recap. So predictions must be verified by live cell assays, like CETSA and nanobret and CRISPR techniques. Then, patient-derived organoids act as 3D test subjects. And finally, MIDD models optimal dosing for safe clinical translation.
Spot on.
It's just fascinating that we can grow a mini version of your tissue to test drugs. For you listening, which part of this closed loop do you think is the hardest to get right? Because to me, imagine if an AI tried to map your daily life with that level of precision, finding the one specific combination of sleep, diet, and stress that triggers a cascade of productivity. It's staggering.
It is.
But for all the success stories, the paper dedicates a massive section to the bottlenecks. AI is only as good as its data, and biological data is notoriously messy. What happens when the garbage in, garbage out principle applies to life-saving medicine?
It creates significant hurdles, to be honest. The first major bottleneck is label noise and assay heterogeneity.
Oh.
Remember, this data comes from thousands of different labs around the world.
Right, they aren't all perfectly identical.
Exactly. A lab in Boston and a lab in Tokyo might run the exact same test on the same protein and get slightly different results due to temperature, region quality, or just human error. If you feed conflicting noisy data into an AI, the model's predictions degrade rapidly.
And beyond just messy data, the source mentions something called shortcut learning or overfitting.
Shortcut learning is a massive problem in AI.
Yeah.
The algorithm suffers from popularity bias.
So the AI is like a lazy student. Instead of doing the complex high-dimensional math to find a novel cure, it just Googles what proteins are famous and guesses those.
That is exactly what happens. The AI scans the literature in the knowledge graph, sees that a specific protein like P53 has been cited in 10,000 papers, and essentially says, well, P53 is popular, so I'll predict that as the target.
Wow.
It completely ignores true, undiscovered biological links because choosing the famous protein is the mathematical path of least resistance. And this leads to another issue, low plausibility.
Meaning the math works, but the biology doesn't.
Yes. The AI might suggest a drug-disease connection that looks beautifully close in vector space, but is biologically impossible because the AI ignored basic pathway logic, like a drug needing to cross the blood-brain barrier.
So how do researchers force this lazy student to show its work and prove it isn't just hallucinating impossible biology?
The field is rapidly shifting toward XAI, or explainable AI. For a long time, AI in drug discovery was a black box. The algorithm spit out a chemical structure and essentially said, trust me.
Which scientists hate.
Absolutely. XAI forces the model to trace its logic back through the knowledge graph, explicitly highlighting the causal steps and biological pathways it used to reach its conclusion.
Which allows human experts to verify the biology before spending millions of dollars in a wet lab.
Exactly. And to fix the data issues, researchers are enforcing strict owed out-of-distribution testing splits.
What does that mean in practice?
It forces the AI to predict targets on completely novel, unseen data, proving it can actually generalize biology rather than just memorizing old, popular data. They're also rallying around fair metadata principles,
Fair.
ensuring that all biological data fed into these models is findable, accessible, interoperable, and reusable.
Okay, but enforcing data standards implies that everyone is actually sharing their data. To build the ultimate biological knowledge graph, wouldn't we need all the major pharmaceutical companies to pool their private, highly lucrative patient and clinical trial data? The financial incentives just don't align for that kind of cooperation.
It's a massive hurdle, not just for corporate IP, but for patient privacy. You can't just upload millions of sensitive patient genomes to a centralized public server. The solution the paper highlights is federated learning.
How does federated learning bypass the privacy and IP issues?
It allows multiple pharma companies and hospitals to train a shared, central AI model without ever moving their private data. The central AI sends its algorithm to the local servers at Pfizer, Novartis, or Mayo Clinic.
Oh, so the algorithm travels, not the data.
Exactly. The algorithm trains on their private, decentralized data locally. Then, the algorithm only sends the learned parameters, the insights and mathematical weights, back to the central model. The raw, private data never leaves the local firewall.
That is fascinating. The insights are shared, but the raw data remains totally locked down.
And the paper even points to the future of this. Quantum federated learning. As these models grow to trillions of parameters, classical computers will struggle to optimize the encryption and the math simultaneously. Quantum computing will eventually be used to securely optimize parameter transfer across these massive decentralized networks.
Which brings us to the timeline. Let's recap these final points real quick. AI suffers from popularity bias and shortcut learning. Solutions include explainable AI, fair data, and privacy-preserving federated learning. So, question for the audience. Would you trust a multi-target drug designed by an AI if the AI couldn't explain exactly how it made its decision? Something to think about, because the article actually lays out a 10-year roadmap for all of this. From 2025 to 2027, the focus is entirely on standardization, getting those fair data principles and explainable AI initiative locked in so we can trust the models. Then, moving into 2028 to 2030, we enter the era of multi-scale personalization. This is where we see fully realized digital twins.
That is the real inflection point. A digital twin is a comprehensive mathematical model of a single patient's specific biological network. Once you combine federated learning with digital twins, you can theoretically simulate how a patient will respond to a multi-target drug before they ever take a single pill.
And finally, the roadmap predicts that by 2031 to 2035, we will see full regulatory adoption. AI-driven network modulating polypharmacology won't just be an experimental technology. It will be the standard FDA-approved method for drug design.
It is a phenomenal trajectory. We are witnessing the evolution of medicine from descriptive biology, where we just observe and try to block what's broken, to predictive design-led medicine. We are no longer just observing the network. We're learning to conduct it like a symphony.
We've gone on an incredible journey today. We started by tearing down the naive hope of the single-target silver bullet, exposing biology for the dense, compensating web that it truly is.
Yes, the interactome.
We unpacked how algorithms use teleporting random walks and geometric embedding spaces to map that chaos, and how those mathematical hallucinations are violently tested in the real world, heated up in thermal shift assays, mutated by CRISPR, and crashed into patient-derived organoids to prove they actually work.
And we confronted the realities of the technology, too. Popularity bias, messy data, and the absolute necessity of explainable AI and federated learning to keep the science honest and secure.
Which leaves me with one final provocative thought for you to mull over. We talked about how, by 2030, this roadmap leads to perfect digital twins. If, in a few years, we have a mathematically flawless digital replica of your personal biological network, and quantum AI can predict exactly how your specific interactome will react to any multi-target chemical combination, does the fundamental definition of disease change?
It's a profound question. If disease is completely predictable, it shifts from being a biological tragedy to simply being an unbalanced equation.
Exactly. Will we stop seeing illness as this terrifying, mysterious force of nature and instead view it simply as an engineering problem, just an unbalanced network waiting for the right algorithm to solve it? It completely reframes what it means to be sick and what it means to be healed. Thank you so much for joining us on this deep dive. Keep questioning the simple answers and keep exploring the complex web of the world around you. We'll catch you next time.